There it is. It's part 3 now, the blood vessels of the cardiovascular system, page 696. And we're back at the table here comparing the artery, the two different kinds of artery elastic, and the, I'm going to flip back here for a few pages just to capture a particular picture that's important. Here we are. It's this portion of the aorta that is elastic, and it's possibly the first half inch or so of these first primary branches. So we have an elastic unit right here that for, your 80 or 90 years of life outside of the womb, you're going through this amazing elastic change with every single heartbeat. So that's quite a system that we've got in place for that purpose. Okay. So we're back now and we're comparing again. When we get down to the arteriole, we've just looked at the elastic artery. That purpose is to maintain blood flow. What is the purpose of the muscular artery? The muscular artery's purpose now is distribution. So rather than having any sort of recoil quality to it, it's simply distributing the blood flow as it's received from the aorta. And we're going to be going then to the various tissues of the body, each muscle organ, the kidney, the lungs, wherever we're going, the digestive system, and so on. Then when we need to look at a particular tissue specifically, we now adjust everything, and we're going to be going now to a arteriole. The arteriole is a very much diminished version of this. It does still have an elastic lamina, so there is some elastic left here. Okay, very little, just enough. Then we get to the capillary, and here we have nothing at all except for endothelium. So that's the diffusion, how shall I say, barrier here to get into the blood is nothing at all. Or I mean to get from the blood into the interstitium, or for the interstitium into the blood. The only thing that's there is the simple squamous epithelial cell plus the basement membrane. Okay, and then we come back up into the venous system, and again we have no elasticity here. Very little elasticity in the walls of the major vascular venous vessels. Okay, so this is our format. We would like to identify, in general, 3 different types of capillaries. We have something here called a continuous capillary, which is what most of the capillaries are. They have occasional small little features that allow for nutrients to flow. Very nice in the way for gases to escape. Occasionally we're going to be having channels in this system here. And in particular, we could have a tight junction. Some of these are tighter than others. So there is a blood-brain barrier. There is a testes blood barrier. So there are certain tissues, the male testes and the human brain in general, have some tight junctions that prevent most of the blood flow. So we actually have a very, how shall I say, nuanced selection of nutrients that make it to the brain and to the testes. Otherwise, most of the other tissues allow for the flow of nutrients. And there are some little pores and clefts and things like that in which nutrients are permitted to proceed. Okay, so here's a intercellular cleft. So we have a little small opening there so that fluid can proceed. In the kidney, we have a different situation because the capillaries in kidneys are actually between 2 arterioles. Instead of going from an arteriole to a capillary to a venule, in the kidney, there is something called a glomerulus, or a little bunch of capillaries, in which they have these sort of like sprinkler holes in them. You can imagine a garden hose with lots of little holes, and it functions as a sprinkler system, right? And so under pressure, under about 50 millimeters of mercury pressure, this particular capillary is actually going to lose the entire fluid volume of the human body, of the blood of the human body, many times per day. We're going to be actually doing dialysis of our own blood in the kidney. And then there's going to be a portion of the kidney that takes up the essential nutrients and the important constituents, solutes of the blood, including water, the solvent, in some cases, in much the way you would want to take it up in the small intestine. So that's sort of like the life of the kidney is very special because we're actually going to clean our blood at the kidney. All of the contents of the blood, except for the formed cells here or the protein, none of the protein really comes out in that through these openings. So these are called fenestrated capillaries because of the little windows, the fenestrations. Then we have another kind of capillary that's called sinusoidal capillaries, and they can be found in the spleen and the liver. The liver in particular specializes in this type of capillary system, and the cells that form the walls are actually going to be called hepatocytes, and they specialize in sampling the blood and picking up certain nutrients and cleaning up certain toxins. Alcohol is one of those toxins. Anything from the pharmaceutical persuasion is usually metabolized in the kidney. Okay, so we're going to be passing through. We still have endothelial cells. We still have some tight junctions, but then there's these open passageways that prevail. Notice how small a capillary can be. Here's a capillary with 2 cells in circumference. Right, so 2 squamous cells holding hands with each other, and the red blood cell can barely get through some of these openings, and they actually enter into what we call roulette. So 1 cell at a time is giving off its oxygen, and then there's also nutrients. The same thing happens back at the lungs, in which the red blood cells are passing by the alveoli where the fresh air is, and are picking up oxygen from the air that's inhaled. Okay, and so they're going by 1 at a time, so that the all the hemoglobin that's available here, carrying 4 molecules of oxygen per molecule of hemoglobin, can give off the oxygen just the way you need it. Okay, so let's take a look here. Here's a capillary network that's fully functional, and the parasympathetic or the sympathetic has engaged the process here so that this particular tissue is being fully utilized. Here we have a tissue. Sometimes we're at odds with one another. If we're sprinting, then this would be the muscles, the way the muscles look, and this is the way the digestive system would look. The digestive system is sort of hunkered down and sort of locked down, as it were, and not really being utilized so much. And this would be the reverse if we were seated at a table enjoying good company and good food. Then we would be having this situation in the digestive system, and we would have less blood flow to the muscular system. And the overall view of where's the blood. And here you can see that most of the blood is actually in the venous system. I would submit to you that possibly more than 60% is there. A very limited amount is actually in the arterial system circulating by the cells. So this is important detail. Over here we have some that's gone off, getting some oxygen over at the pulmonary system. Here we have blood that's actually in the heart or perfusing the heart. And then here we have blood in the capillaries that are actually engaged in the transfer of nutrients and so on. But somebody might ask, what was the position of the body at the time of death? You can always know what that is because the patient stops moving, of course, and there is equilibrium, that thing that only happens once in a lifetime. And all of this coagulates. And you can see bruising on the skin. Whatever is the most lowest part of the body, we call it dependent lividity. Wherever the lowest part of the body is, if your patient has died face up in a hospital bed, then their gluteal region and their shoulder blades will be quite bruised, and that's because the blood has flowed to that particular area. Okay?